Okay, so welcome back to this next video in which we are discussing voltage-gated potassium channels. Okay, right. So in the previous video what I showed you was the structure of the selectivity filter, and I showed you how it is this cage consisting of 4 separate binding sites for potassium, okay, and these 4 binding sites are formed by these 8 corners of each one of these binding sites, which have electronegative oxygen atoms positioned there. Okay, and these electronegative oxygen atoms will have partial negative charges, and it's that partial negative charge that can interact with the positive charge of the potassium. Okay, and the reason that this selectivity filter is selected for allowing potassium to move through it is that only potassium is going to fit there nice and snugly and be able to interact with these 8 corners of the binding site in such a way that the amount of energy that you will release from that binding is going to be greater than the amount of energy you have to put in to pick off the 8 water molecules that make the hydration shell of the potassium ion. Whereas for other ions, the amount of energy that you have to put in to remove the hydration shell will be greater than the amount of energy you'll get out by it interacting with these 8 corners of the binding site within this selectivity filter cage. Okay, right. So now what I want to show you is how potassium ions actually move through the selectivity filter. I've shown you how a potassium ion can bind to one of these binding sites of the selectivity filter, but that doesn't show us how they actually end up moving through it. Okay, so let's go over the page and I'll show this. So, let me draw now the selectivity filter from one side. Okay, so here it is, with its 4 separate binding sites for potassium shown here. Okay, and let's say that this is the intracellular side and this is the extracellular side. So putting it in the context of the larger potassium channel, we'd have it like this. Okay, so this is the pore-forming domain of the pore-forming portion of the potassium channel. Okay, so here we go. Here is the selectivity filter here. This is the intracellular aspect. This is the intracellular part of the pore, which remember is much wider. This is the P-loop here, lining the edge of the selectivity filter, so I'll put that in vivid purple here. And then behind that you'll then have the S6 membrane spanning alpha helix, shown here in blue, and then behind that you would have the S5 membrane spanning alpha helix. And clearly the channel at the moment is in the open state because the S6 helices are not obscuring the intracellular aspect of the pore. Okay, so remember when you open these voltage-gated potassium channels, potassium is going to move out of the cell. Okay, so what's going to happen is a potassium ion is going to come in from the intracellular aspect here. Okay, it's going to shed its hydration shell and then it's going to go into the first binding site, like so. Okay, so here is our potassium ion in the first binding site. Then what's going to happen is another potassium ion is going to come up behind this towards this first binding site. And remember, potassium ions both have positive charges. So this new potassium ion that's coming up here, this is going to push this potassium ion here from the first binding site into the second binding site. Okay, so this potassium ion is going to end up moving to the second binding site here. Okay. Then what's going to happen is this potassium ion here, the new one, so this was potassium ion number 2, so I'll colour in potassium ion number 2 in a separate colour. Potassium ion number 2 is then going to move into this binding site here, the first binding site, okay, like so, and when it moves into the first binding site, it will then repel potassium ion number 1 from the second binding site into the third binding site here. So now potassium ion number 1 will move even further along. Okay, so now, at the end of this, what we've now got is this situation here. If I just draw the 4 binding sites of the selectivity filter now, here is potassium ion number 1, and here is potassium ion number 2. Okay, so this is potassium ion number 1 in red here, and this is potassium ion number 2. Okay, and then all that needs to happen now is potassium ion number 3 needs to come along, which we'll show in blue here. it will then do exactly the same as potassium ion number 2 did to potassium ion number 1. Okay, so it will come over here, and then potassium ion number 2 will get pushed into the second binding site, and when potassium ion number 2 moves into the second binding site here, what will then happen is potassium ion number 1 will have to move from the second binding site into this fourth one up here, like so. And then, what's now going to happen is that potassium ion number 3 here is going to move into binding site number 1, and then number 2 will have to move out of there into number 3, and then number 1 will have to move out totally. Okay, so what's going to end up happening is you're then going to go to this situation. So if I draw it again, you'll have potassium ion number 2 now in that one, potassium ion number 3 in this one down here. So there's potassium ion number 3 in blue, here's potassium ion number 2 in green, and then potassium ion number 1 will now have been pushed out to the extracellular side, and then it can continue on. Now potassium ion number 4 can come in, and this can continue happening. You'll continually get potassium ions being moved through the selectivity filter by other potassium ions coming up behind them, basically, and repelling them away. Okay, so that is the mechanism of permeation of potassium ions through this selectivity filter. Okay, right. What I now want to turn our attention to is the process of inactivation. Okay, so let's just summarize what we've got to so far. We started off with this closed voltage-gated potassium channel here, and again I will just show the pore portion at the moment. Okay, so here is the closed potassium channel here, okay, where the S6 helix was blocking the intracellular aspect of the pore. Then when you depolarize the electrical potential difference across the cell membrane, activation occurred. Okay, remember the voltage sensing domains moved outwards. This conformational change was conveyed to the pore forming domains via the S4, S5 linker, and that pulled the S6 helices out of the intracellular aspect of the pore to open it up. Okay, so then you went into the open state here. Okay, where the pore is now open and conducting potassium ions. Okay, so this is the open state. Now, if you then repolarize the electrical potential difference across the cell membrane, you can turn it back to this state. And that process of closing the voltage-gated potassium channel by repolarizing the electrical potential difference across the cell membrane, that's known as deactivation, not inactivation. Okay, inactivation is the process by which this open channel can close even though the electrical potential difference across the cell membrane is still depolarized. Okay, so something else is going to change basically. What's going to end up happening is something is going to come in and block this intracellular aspect of the pore. So let me show this here. So I'll just move this up a little bit. Okay, I want them all in view. Okay, so now what you're still going to have is the S6 helix nicely moved out of the way here. Okay, but now you're going to have something new coming in here and blocking the channel. Okay, so what is this thing that can come in and block the channel here? Well, there are different possibilities for what this can be. Okay, it can be part of the amino terminus of one of the alpha subunits of the voltage-gated potassium channel. Okay, so let me draw this out again. So if I draw an alpha subunit of a voltage-gated potassium channel once again, okay, so here is the tetramization domain here, T1. Then here is the amino terminus portion here. Then we have S1, S2, S3, S4, the S4, S5 linker, S5, the P loop, and then S6. Okay, and then the C terminus. Well, remember the voltage-gated potassium channel is made up of 4 of these structures. Basically, what can happen in inactivation is one of these amino terminal portions, and it's not totally understood which portion it is. One of these amino terminal portions can come in and block the pore from the intracellular aspect. Okay, and it could be one of the amino terminal portions from any of the 4 subunits of the voltage-gated potassium channel. You have 4, remember. Okay, so potentially you could have 4 of these amino terminal portions that are capable of coming in. Okay, it's not known whether this is something that can happen for every single possible alpha subunit of a voltage-gated potassium channel. Okay, there are certain alpha subunits of voltage-gated potassium channels which do seem to have an amino terminal portion which is then capable of coming into the pore from the intracellular aspect and blocking it, and then blocking any potassium ions from moving through. Okay, this is why the process is known as N-type inactivation. Okay, because it's referring to the amino terminal portion in this name. Okay, N stands for the amino terminus portion here. The amino terminus portion is blocking the pore, and that's how you're going into this inactivated state. Okay, so this is also referred to as the N gate, the way of shutting the pore of the potassium channel using a different gate to the V gate. This was the V gate here. The N gate is this gate on the amino terminus of the subunits of the voltage-gated potassium channel. Okay. In addition, in some cases, it doesn't actually seem that this has to be a portion of the amino terminus of one of the alpha subunits of the voltage-gated potassium channel. In some cases, it seems that it can be part of the beta subunits. Okay, so if I get my picture back again, here is my picture of the entire voltage-gated potassium channel here. Okay, we're saying that some part of the amino terminus of one of the 4 subunits can come in and block the pore, but in fact it can also be part of one of the beta subunits, okay, that can come in and block the pore, and that's still counted as N-type inactivation. Okay, so this then is called a ball and chain mechanism. Okay, so the way that you're getting this ball coming into the pore and blocking it, which is on some sort of chain like so, that's why it's called a ball and chain mechanism, and you'll find a very similar mechanism underlying the inactivation of voltage-gated sodium channels. Okay, so ball and chain mechanisms have a big place in molecular biology. Okay, right. So this process here, where you are closing the channel even though the voltage across the cell membrane is still depolarized, that's known as inactivation. Okay, now, if you then repolarize the electrical potential difference across the cell membrane, what you want to do is go back from being in this inactivated state here. This is not called a closed state, this is an inactivated state. You want to go back from being in the inactivated state to being in the closed state. Well, the first thing that has to happen is you have to temporarily go back to being in the open state. Okay, you have to firstly remove this structure that is obscuring the pore from the intracellular aspect. You have to reopen the N gate, as it were. Okay, that process is known as de-inactivation. Okay, so removing the inactivation gate, removing the N gate, opening the N gate again, is the first thing you have to do if you want to go back to the closed state. So you remove the N gate, you remove this obstruction. That temporarily takes you back into the open state, but now, because we're back in the repolarized phase, the instant you remove that obstruction, what will happen is you will return back to this conformation and you'll once again be in the closed state. So, to go from the inactivated state to the closed state, you have to temporarily go through the open state. So you have to go through de-inactivation and then deactivation. Okay, right. So that then is the the N-type inactivation of voltage-gated potassium channels. There are new types of inactivation being discovered, for instance C-type inactivation, but there is very little known about C-type inactivation, which is why I'm only going to mention that it exists. Okay, so that concludes then our discussion of voltage-gated potassium channels. We have seen the structure of voltage-gated potassium channels. We've seen how they are activated by depolarization of the electrical potential difference across the cell membrane. We've then seen how they actually allow potassium ions to move from the cytoplasmic side of the membrane to the extracellular aspect of the membrane, and now we've seen how they can inactivate even when the electrical potential difference across the cell membrane is still depolarized. Okay, and then we've seen to reverse that inactivation, you firstly have to go back to the open state temporarily, and then you'll go back to being in the closed state.